This will be a walkthrough of the AQA GCSE Biology 2022 Higher Tier Paper 2. Question 1. There are two types of reproduction: sexual reproduction, asexual reproduction. 1.1. Complete Table 1 to compare sexual reproduction with asexual reproduction. Write a tick in the box if the statement is true. The first row has been completed for you. So we need to remind ourselves what is involved in sexual reproduction and how is asexual reproduction different and similar. So as you can see already, they've told us that they're both examples of cell division, which is why both boxes are ticked. However, sexual reproduction is actually fusion of gametes, whereas asexual reproduction is where you are making clones, so they are genetically identical. So if you've got gametes, which are your sex cells, they need to fuse, which is called fertilization. So sexual reproduction involves fertilization. It does not occur in asexual reproduction. Next one, genes are passed on from parent to offspring. Well, the genes are going to be carried in the gametes, which would mean that the offspring should also have those genes, so that needs to be a tick. And if it's asexual reproduction and you're making clones, they're genetically identical to the parent, so they must have the exact same genes as the parent, so this should be a tick too. Offspring are genetically identical to each other. We just said that term multiple times now. That's only in asexual reproduction, which is why this one should be ticked only. Cell division by meiosis forms gametes. Figure 1 shows the mean mass of DNA per cell before, during, and after meiosis. Use information from Figure 1 to answer questions 1.3 to 1.6. So looking at the graph now, we can see that the mass of DNA per cell stays at 4 for about 3 days, then increases to 8, so that's doubling there. Remains at 8 for a day, then drops back down to 4, so that's halving there. Then remains at 4 for a day and then drops to 2, so that's another halving there. And then remains at 2 for the rest of the time. When is the DNA in the chromosomes being copied? Tick one box. So if it's being copied, we should expect it to double, so multiply by 2. So in which time period is that happening? So look at our graph again, which we've annotated. That time when it's multiplying by 2 is between 3 and 4 hours. Cells divide twice during meiosis, which 2 times in Figure 1 show one cell dividing into 2 cells. Well, if it's one cell dividing into 2 cells, we should expect the mass of the DNA in the cell here to half, so drop by 2. So we're looking now for those moments, because it says 2 times, because we need to tick 2 boxes, 2 times when it's dropping by 2. So we can see at this point here, 5 hours, it's dropping by 2. You can see at 6 hours, it's dropping by 2 again also. So our 2 times which we're going to tick are 5 hours and 6 hours. 1.5. What is the mean mass of DNA in arbitrary units in a sperm cell? Okay, so we're applying our knowledge right now in this question. A sperm cell is a gamete. So if it's a gamete, what does the gamete want to do? It wants to fuse with another gamete. So in this case, the sperm wants to fuse with an egg cell. So we should expect then our DNA in our sperm cell or gamete to be half, and our egg cell to be half, and then when they come together, then they'll have a full set. So let's have a look then, back at our graph. When do we expect it to be at this half period? Well, we've got 2 times when it's half. But here is the thing now. In meiosis, it's 2 cell divisions that are occurring. So our meiosis would really be from 8 to 4 to 2. So at the end of meiosis, we get our gametes produced. So our gametes that we're producing here should really have 2 in terms of its mass. So going back to the question now, a mean mass of DNA in a sperm cell needs to be 2 in terms of its mass. What is the mean mass of DNA in arbitrary units in each cell in an embryo? Well, our embryo then is going to be produced as a result of fertilization. So when the sperm and the egg have fused. So at that point then, it would need to be double what is in a gamete. So if we remember it again, our gamete was supposed to have a mass of 2. So here was our sperm, and also our egg would be the same. So when those 2 come together, it will be doubled, so it will be 4. So we should expect our mass of DNA in our embryo to be 4 arbitrary units. Question 2. Earthworms live in soil, feed on dead and decaying plant matter, have soft, moist skin, exchange gases through their skin. Give 2 abiotic factors and 2 biotic factors that could affect the size of an earthworm population. Right, so the only way that we can answer this question properly is if we recall what these 2 terms mean, abiotic and biotic. Well, abiotic means non-living factors that can affect an ecosystem. Biotic factors are living factors which can affect an ecosystem. So we've got to think now, what is something that is non-living in an ecosystem that would be able to affect a population of earthworms? Well, an earthworm is an animal. So something that's non-living that will affect an animal is definitely going to be oxygen levels. So levels of oxygen can of course cause there to be either more or less earthworms. Now, looking back at our points here, because these bullet points are important information for us at some point. So you can also think about what from here might indicate to us something that might be an abiotic factor. Well, it says they have soft, moist skin. So if there's moist skin, it's got to do with water. And the amount of water that could be present can definitely affect an earthworm population. Now, if there's water that's present in the air, that is known as humidity. So the levels of humidity that may be present in the ecosystem could affect our earthworm population. Because if there's low humidity, then their skin's not going to be moist, and then that could cause them to die. Biotic factors. So what may be living that may affect the amount of population of earthworms? Well, earthworms are part of a food chain or food web, so they are unfortunately going to be preyed upon by predators. So the presence of predators is going to be a factor. If there are more predators, that means that there will be less of our earthworms. But earthworms are animals. They're not at the bottom of the food chain. They're not at the first trophic level, so they're not producers. So they have to feed too. So it's their availability of food for them, which is also going to affect how much is present for them. 2.2. Students investigated the populations of earthworms in the soil in two different areas. Area A, a grass lawn. Area B, a farmer's field. Chemical X can be mixed with water and poured onto the soil. The mixture brings earthworms to the surface of the soil, but does not harm the earthworms. Plan an investigation using Chemical X to compare the number of earthworms per meter squared in areas A and B. Okay, so this is our example where we have to use our knowledge from our required practical of a field investigation. So there must have been a time where you have studied how you could either measure the abundance of a particular species, whether it's plant or animal, or the distribution of a particular species of plant or animal. Now, depending on which method you are looking at, there is a different way to do it. So we need to think now, which one of these methods do we need to talk about here in order to satisfy the 6 marks that this question is offering? Well, the first thing that we want to look at to figure out which method to go down is if there is a change in a factor. Well, in this case, the factor is chemical X. But we're not looking at how chemical X changes from one place to the next in a certain area. We're looking at two different areas, and we want to compare the number of earthworms. So, because we're not looking at how it changes over a specific place, we're not investigating distribution. We are investigating abundance. So, when we're investigating abundance, then there are two very, very important things which we need to include. The first thing is it needs to be random. So that means that we have to be able to count the number of earthworms, but in a random manner. So we're trying to avoid bias. The second thing that we need to include is the piece of equipment that we're going to use to do this, and it will be a quadrat. So, discussing the fact of doing this randomly somehow, and discussing the fact that we need to use a quadrat is going to allow us to pick up some marks, definitely. So, the first thing that we need to think about then is we need to place this chemical X on the field. And of course, we're looking at two areas, so we need to place this chemical X in both areas, area A and B. Now, remember that we're doing an investigation, an experiment here. So, for us to be able to compare these two different areas, we need to keep some things the same. Now, as it says, chemical X can be mixed with water and poured onto the soil. So, if it's a chemical, a liquid, there are some things that we probably want to keep the same about this liquid to ensure that we can compare these two areas effectively. And the two obvious things for any liquid are the concentration and the volume. So, what we need to say is the concentration and volume of chemical X should be the same when applied to both fields. Okay, so we discussed now about our chemical X. Now let's discuss this randomness. So to ensure that there is no bias, we want to be able to put our quadrat down on our areas in a random manner. So the best way for us to do this is to create a grid of our field. And once we create a grid, we're able to then plot coordinates. Now, what does this actually mean? It means if this is our field, what we can do is we can split our field into sections to create a grid. And what that allows us to do is to essentially have an X and a Y axis for our grid of our field. And then each of these squares can be assigned a number. So this could be 0, 1, 2, 3, 4 for our x-axis, and then 0, 1, 2, 3, 4, 5 for our y-axis. And then what we're able to do with this information now is we're able to randomly choose places on this grid, which translate to our area on our field, to place our quadrat. So the best way for us to do this, still being random, is using a random number generator. And then this will determine where to place our quadrat. Remember, we should not be throwing a quadrat down, so any indication of trying to throw quadrat over our shoulder should not be discussed. It's better to discuss this type of method using a random number generator. Okay, so once we've placed our quadrat down, what we're then going to do is count the number of earthworms in the quadrat. Okay, great. So we've done that. However, this count that we do could really be lucky for us, because it may be on the off chance that we enabled a lot of earthworms to appear. So what we definitely want to do is we want to do this multiple times. So repeat at least 10 times. So we have a good representation of our area. Okay, so we repeat it at least 10 times and then repeat for area B. Okay, so once we've repeated 10 times on both areas, the next thing that we should be thinking about doing, because we've done repeats to ensure that there's no anomalies in our data, is calculate a mean. Okay, fantastic. And once you've calculated a mean, you've then got a mean number of earthworms for area A. We've got a mean number of earthworms for area B. We're then able to just compare means to identify which area has more earthworms. What we could actually do to compare means better is we could do something like drawing a bar chart, because I would visually be able to see which one is greater. Question 3. It is important to control the concentration of glucose in the blood. Figure 2 shows how the concentration of glucose in the blood of a person changed over 4 hours. So looking at our graph, we can see that the concentration remains steady, just about 4.3, 4.35, and then just after 1 hour it increases up to about 6 millimoles per decimeter cubed, then decreases for about 2.5 hours, then after that increases, and then remains constant up until 4 hours. Specifically, there is a time point on our graph where a meal is eaten. So 3.1, give one time when the concentration of insulin in the person's blood would be high. So what is our insulin? Need to remember what that is now. It is a hormone released from the pancreas. And what does this hormone do? It detects a high concentration of glucose in the blood and causes it to be lowered. So what we're expecting then is this insulin to be high when the concentration of glucose is high. So when there's a concentration of glucose high, we need a time period, not a blood glucose concentration. So any time, the highest point would be around here. So if we were to read from our peak of our graph, it would be around 1.6 or 1.7 hours. And that would be a suitable time to use. I would assume at any time just after the meal to before the concentration gets too low of glucose, below the normal level, would be fine. Three point two. Explain the effect a high concentration of insulin has on blood glucose concentration. Well, as I said before, the insulin is responding to a high concentration of blood glucose. So when insulin is higher, it's going to cause the blood glucose concentration to decrease. So when glucose is increased, the pancreas releases insulin. So our effect of this high concentration of blood glucose is going to be that insulin is going to decrease it. So it's going to be lowered. But what is actually going to be the reason why it's going to be lowered? Well, we have to remember what happens to this glucose when insulin is released. The glucose is a carbohydrate, but it's a soluble carbohydrate. So it gets converted into an insoluble carbohydrate, and that is called glycogen. So this storage of carbohydrate now can't be dissolved in your blood. So what's going to happen to it now is it's going to get stored, and it gets stored in two places: stored in the liver or stored in muscle cells for it to be used later. So that's what we need to say now. The glucose is converted to glycogen and it's stored in the liver and muscle cells. And that should be our 3 marks. People with diabetes have difficulty controlling the concentration of glucose in their blood. Type 2 diabetes is linked to obesity. Figure 3 shows how to find if an adult's body mass is healthy for their height. Okay, so we've got these different categories of weight and the relationship between the body mass and the height here. 3.3. Person A is 1.75 meters in height, has a body mass of 52 kilograms. What is person A's weight category? So we've got to go back to the graph and look for those specific values to find out what category they're in. So 52. So 52 should just be around here on our graph. And we're looking for 1.75. So 1.75 should be around halfway between them. So if we connect those 2 points on our graph, the person's category will be underweight. 3.4. Person B is 1.9 meters in height. Give the range of body masses that would put person B in the healthy weight category. So this time we're looking for 1.9, but we need to stay in the healthy weight category now. So 1.9, but we need to stay in the healthy weight category. So healthy weight starts there, and then it ends there. So remember, we need the masses now. So going down on this line, that's going to be 90 kilograms. But where is it going to start? It's going to start somewhere just down here. So that's just below 70 kilograms. So we could say it's roughly around 67 to 68 kilograms for there to be in the healthy weight category. So let's just be safe and put 68 to 90 kilograms. 3.5. Person C is obese. A doctor thinks that person C has type 2 diabetes. The doctor tests a sample of blood from person C. The results of the blood test is shown in table 2, and also the mean results for people who do not have diabetes is shown in table 2. Right, so what have they told us then? It's also given us the cholesterol concentration, the glucose concentration, and the concentration of insulin that's going to be in the person's blood. Type 2 diabetes occurs when body cells have a reduced response to insulin. Oh, that's great. So they've reminded us now what type 2 diabetes is. We should know this though. Give 2 ways the results of the blood test show that person C might have type 2 diabetes. Well, if the body cells have a reduced response to insulin, that should mean that the insulin concentration will be very, very high. So, looking at our table now, person C has a very, very high concentration of insulin in their blood compared to the mean of those that don't have it. So that would be our first way of telling. The other thing that we should expect then, because it's type 2 diabetes, diabetes is when you've got a higher concentration of glucose in the blood, would be that higher concentration of glucose, again compared to the mean. So remember, what we're doing here now is we need to, in our answer, show a comparison, because they've given us a person C now, so it'd have to be something that person C has, which indicates that they may be suffering from type 2 diabetes. So the first thing we said was a higher concentration of insulin, and we then said a higher concentration of glucose. And those are our two ways. Three point six, give two ways that a person can reduce the chance of developing type 2 diabetes. So, how can you treat type 2 diabetes, or how do you prevent someone from developing it? The most common way, which should be said, is with regular exercise. For someone with type 2, though, the problem is again is that the body is not responding to this insulin that is being produced by the pancreas. So the one way to combat that then is to stop the glucose concentration being high in the blood. And other than exercise, the other way in which that can be addressed is by reducing the intake of carbohydrates. So what you'd want to say then is either eating less carbohydrate, or you could say have a carbohydrate controlled diet. So that I'm assuming would be the same as this point too. The rapid growth in human population means that more waste substances are released into the environment. The release of substances into the environment can cause pollution. Name one harmful substance that could cause air pollution. So again, we're thinking about what could be in the air that could be a problem. So our most common thing that could be in the air, gas, would be carbon dioxide. 4.2. Name three harmful substances that could cause water pollution. Do not refer to plastic or litter in your answer. So we're thinking about something that could go into the water, and if it's talking about pollution, it's going to cause some problems. So a common thing would be to think when thinking about pollution is what could kill any aquatic organisms. So for example, fish or aquatic plants, pondweed. So what could cause that to happen? Well, a very common thing is any fertilizer, because fertilizer should be only on land. However, it can leak out into water and then cause a big problem such as eutrophication. Another thing that could be in water which shouldn't be there is oil. There's been a lot of cases where there have been massive oil spills, and then any wildlife that's been in water can be affected by that, killed by it. And another thing that we could mention could be an example of a chemical such as sulfur dioxide. Even though sulfur dioxide can lead to acid rain, we can think of it as air pollution. But of course, if the sulfur dioxide then goes into the water as part of the acid rain, that can also kill any aquatic organism which may be there too. 4.3. Describe how substances that pollute air and water could be harmful to humans and other living organisms. 6 marks. So right now then, we're looking at specifically what does air pollution do, what does water pollution do. So we have to go into a lot of detail here in order to try and get our 6 marks. So if we start with air pollution then, again, we gave an example in the previous question, is carbon dioxide. But what are some other examples of things that can contribute to air pollution? Carbon monoxide can do that. You said as well, sulfur dioxide could do that. So what we want to do now is you want to remind ourselves of what are the effects of carbon dioxide as part of air pollution. Well, it leads to global warming. And then what does global warming lead to? Well, it leads to climate change, but what are the real effects of those? Real effects of climate change is melting of polar ice caps. So we need to give those specific examples. It can lead to flooding. Can lead to drought. And then it can lead to change in migration patterns of animals such as birds. Carbon monoxide, well, that's a poisonous gas. So what is going to be the problem with carbon monoxide? Well, carbon monoxide leads to breathing problems because you're not able to carry as much oxygen anymore. So it's less oxygen that's being carried. And then that can lead to respiratory problems, such as asthma. Sulfur dioxide. Well, we know that that can lead to acid rain. But what is the problem with acid rain now? That is going to damage plants. So if plants are damaged now, then that means if it's the leaf, it could lead to less photosynthesis. So we've spoken about carbon dioxide, carbon monoxide, sulfur dioxide. We've spoken about the actual effects of them now. We now want to then think about water pollution and the problems there. So specifically now, fertilizer. So we said that before. And what's the problem with fertilizer? Well, it's leading to this issue called eutrophication. So if we just expand on what eutrophication is, fertilizer leaks out into nearby rivers and streams, and then it causes something called an algal bloom, where the algae reproduce very quickly. And what does this algae reproducing do? It blocks the light from entering that area of water, so that leads to less photosynthesis of any plants that are in the water. And then the consequence of that now is these plants are going to die. And if these plants die, then two things will happen. One, they won't be producing any oxygen, so there'll be less oxygen in the water. And two, they're going to be decomposed and then broken down. And these decomposers that are breaking down these dead plants, they're going to be using any oxygen. So ultimately, there will be less oxygen. And for example, fish then, let's use that example, less oxygen for fish. So they won't be able to carry out aerobic respiration, and therefore they die. I think we need another example now of water pollution. So let's use an example of the sewage or oil, for example. That's an example. So what will be the problem there then? Problem of any sewage or oil is going to then cause there to be a buildup of bacteria. And then what these bacteria are going to do, they're going to respire aerobically. So again, there's less oxygen for any aquatic organisms, so they die. Okay, so it's 6 marks here, so it's not going to be for single points here. We need to try and make sure that we've covered pollution of air, pollution of water, and then talking about how they can be harmful to humans. So we've definitely done that in terms of global warming, drought, because if there's a drought, there'll be less crops grown, so that can cause famine, and other living organisms. So we've spoken about plants and we've spoken about fish. So we've said examples that will be able to satisfy 6 marks here based on the criteria that they're asking for us in the question. Question 5. Maple syrup urine disease, MSUD, is a rare inherited human condition. MSUD is usually diagnosed early in childhood and can be controlled by having a low protein diet. Figure 4 shows the inheritance of MSUD in one family. Okay, so what this is known as is a pedigree diagram, and it allows us to see how an inherited disorder can be passed on through a family, and also allows us to figure out the chances of developing it if a family have another child. The allele for MSUD is recessive. So recessive now means requires 2 alleles to be expressed. It's an important reminder there. Give one piece of evidence from figure 4 which shows that MSUD is a recessive condition. So, as we were saying, it requires 2 alleles to be expressed. So that means that both parents would need to have those alleles for their offspring child to have it. So let's find 3 offspring and their parents particularly. So we can't use 4 because 4 is the parent. We don't have the parents of 4. So our examples that we could use are 5, or we could use 12 here. So what do we notice about 5? 5 has the condition, but their parents don't have the condition. If their parents did have the condition, then 1, they would be darker in terms of the box of the circle, and it would mean then that all of their offspring should have the disorder too. So that would be our way in which we can tell that this condition is recessive. If we look at 12, it's the same situation. 7 and 8, they both don't have the condition, but they give rise to 12, which does. So we can use this example then. Parents 1 and 2 give rise to 5. What we need to make sure that we say though is parents 1 and 2 don't have MSUD. (5.2) Persons 7 and 8 in figure 4 are expecting a 4th child. Determine the probability that the child will have MSUD. You should draw a Punnett square diagram. Identify the phenotype of each offspring genotype. Use the symbols capital N allele for not having MSUD, lowercase n allele for MSUD. Okay, so we have to go to 7 and 8. As we said before, because we can use the information from the previous question to help us now, because we could have used 12 as an example in 5.1, but we didn't. We used 1, 2, and 5. But 12 has the condition. So if we're using the letters from 5.2, the genotype of 12 would be n lowercase n lowercase. Remember that genotype is the combination of alleles. Phenotype is the physical expression of the genotype, the physical expression of this combination of alleles. So with these two lowercase n's, we get this male with MSUD. But 7 and 8 don't have the condition. But for them to give rise to 12, it must mean that each one of them is carrying an allele for this condition. So that means that 8 would have to have one lowercase n for it to be passed on to 12. But it's not showing the condition, so that must mean that it must have a dominant allele, capital N. Seven, again, is the same principle. They have one lowercase n, which can go to 12, but they must have one dominant allele because they're not displaying the condition. So both 7 and 8 actually have the same genotype, and when they have different alleles, we classify this as heterozygous. So 7 and 8 are heterozygous. So what we need to do now is we need to create a Punnett square diagram, because that's what they're asking us to do, to show the probability of a fourth child getting this condition. So we're going to draw our Punnett square now. So our Punnett square is a grid, and we can put our offspring genotypes in our squares here, but our parental genotypes need to go at the top and on the side. So if we put the father here and the mother here, remember they're both going to have the same genotype, so it'll be capital N, lowercase n, capital N, lowercase n. So what we need to do now is do the combinations for all four of them. So in this box here, it'll be capital N, capital N. In this box here, it'll be a combination of these two. Remember, capital should always go first, dominant allele. So capital N, lowercase n. In this one, it'll be the same, capital N, lowercase n. And then this will be our lowercase n. So we've done our Punnett square diagram now. We need to identify the phenotype of each offspring genotype. So remember, we've done this already. So this double recessive genotype will be has MSUD. This one not MSUD. Again, should be the same here, not MSUD. And then this one, because it doesn't have any lowercase alleles at all, definitely not MSUD. So there is only one offspring out of the 4 potential offsprings that 7 and 8 could have, which has MSUD. So if the total is 4, and there's only one possible chance of having it, that's our probability, 1 out in 4. But if we want to put it as a decimal, it would be 0.25, and if you were to put it as a percentage, it would be 25%. Figure 5 shows chemical reactions involved in the normal breakdown of some types of amino acid inside body cells. Okay, so we have some amino acid, it says in this box. And then when enzyme 1 is present, two things can happen. We can have toxic substance P, which then is acted on by enzyme 2, which gives us harmless products. Or we could have enzyme 1 creating ammonia, and then acted on by other enzymes gives us urea. A person with MSUD cannot make enzyme 2. Okay. 5.3. One of the final products shown in figure 5 is urea. Where in the human body are the reactions shown in figure 5 most likely to occur? Okay, so 5.3, we're looking for this place in terms of where ammonia, which is converted by these other enzymes into urea, where in the body is that happening? Well, it's not happening in the small intestine, because in the small intestine we're just getting absorption of any food that we intake into our blood. It's not happening in the pancreas, because the pancreas is where enzymes are released and where hormones, insulin and glucagon are released. The kidney is supposed to be doing two things: filtering the blood and producing urine, which should contain urea, but this conversion of the ammonia to urea is only going to happen at the liver, because that's the process of deamination which occurs in the liver. Scientists can analyze blood samples or urine samples to see if a person has MSUD. The test identifies high concentrations of toxic substance P shown in figure 5. Explain why the blood of a person with MSUD will have high concentration of toxic substance P. Use information from figure 5. So let's go back to our figure 5. And we've got to focus on this toxic substance P. It said in the question, why should there be a high amount of this in the blood with someone that has MSUD? Well, underneath the figure, it reminds us a person with MSUD cannot make enzyme 2. So this enzyme 2 is not present. So the enzyme 2 is supposed to convert the toxic substance P into these harmless products, but again, that's not happening because there is no enzyme 2. So what's going to happen to toxic substance P then is that it will just build up and accumulate, so its concentrations will just increase. So that really should be the answer. The enzyme 2 is not present, so cannot convert substance P into harmless products. So if this substance P into harmless products cannot be converted, it means that they're just going to build up. Therefore, toxic substance P builds up. Okay. Now, the thing that we haven't addressed is this blood. They've done this on purpose. They've put this word bold for a reason. We have to think about this now. What's this reason why they've put this in bold? Well, let's go back. Amino acids we're supposed to get from our diet, from consuming proteins. What's supposed to happen to these amino acids? Well, they're supposed to, once they form from proteins, they go to our small intestine and then are absorbed into our blood. So the problem is, though, is that some amino acids is converted into toxic substance P. So it probably means that the same mechanism to get the amino acids into our blood is what's going to cause the toxic substance P to go into our blood. So the question is, how does the amino acids normally get into our blood? They normally get into our blood by diffusion. So if this toxic substance P builds up, then the toxic substance P will then diffuse into our blood. And that would be the reason why there's a high concentration in there. 5.5. Explain why the urine of a person with MSUD will have high concentration of toxic substance P. Right, so it's very similar to 5.4. 5.4 we were speaking about the blood now, and 5.5 we're thinking about urine now. So in 5.4, really the organ that we were thinking about was the small intestine, because as we were saying, that's where the diffusion is going to happen. It's going to happen from the small intestine into the blood. But with urine, it's not going to be now the small intestine that's going to be involved. It's going to be the other organ that's involved. Now remember here in 5.3, we were breaking down the roles of some of these organs. So again, you can see that they've done this on purpose. We've spoken about the small intestine today in this question. We spoke about the fact that diffusion occurs there. We know that the correct answer here is liver. So the other organ that's linked to urine is the kidney, because we said that this is where the urine is produced. So we have to be referring now to the kidney in this answer. So why would we expect to have high concentration of toxic substance P in the urine? Well, what happens at the kidney? At the kidney, you have filtering of your blood. So anything that is in the blood is filtered, passed out of the blood, and then as it goes through the kidney, it's then reabsorbed. Now, the thing is, this reabsorption depends on the size of the molecules. So, small substances will be reabsorbed into the blood. Large substances will not be reabsorbed into the blood. So, what we need to think about then is, why is the urine going to have a high concentration of toxic substance of P? Well, it must mean then that if it's in the urine, then it hasn't been reabsorbed. So not reabsorbed. So what that means then is that the substance P was filtered. So actually passed through any walls in the kidney. But as we're saying now, if there's a high concentration of it, then it was not absorbed back into the blood. 5.6. Explain why a person with MSUD must have a low protein diet. So they did say this before when we were doing our question with the Punnett square. They did say that they need to do this, control this by having a low protein diet. So we need to think about why. So again, protein, we said that that's going to be broken down to form amino acids. And now we've seen from this figure what can happen to some of the amino acids. It could either be converted to ammonia and then urea, or it could go and form this toxic substance P. So what we're trying to avoid then is the buildup of this toxic substance P. And remember, it says toxic. Toxic is damaging. damage into your cells, tissues, and potentially your organs. So that's gonna probably be the last point that we want to think about. The whole point they must have this low protein diet is to prevent damage to cells and tissues. But we've got to make the link now. What's going to be damaging to the cells and tissues? It's going to be this toxic substance P building up. So how do we prevent toxic substance P from building up? Is we have less amino acids in our diet. So that should be the point. We need to just make it clear then that proteins are broken down into amino acids. And then the amino acids need to be at a low amount. And then that's to prevent buildup of toxic substance P, which can damage cells and tissues.